Hi, my name is Kendria Beers. The summer of 2018, I took a break from my Oregonian lifestyle portrayed here to participate in the Data-Intensive Scientific Computing Research Experience for Undergrads program at the University of Notre Dame. Now, imagine you're studying the genetic basis of aging. You'd like to study humans directly, but you're constrained by time and ethical concerns. However, you do have access to a well-studied yeast PPI network, where nodes are proteins and edges are interactions, and you know it partially lines up with a human PPI network due to a shared evolutionary history. You can transfer biological information from the yeast network to the human network. But how do you find this node mapping in the first place? This popular computational problem is called network alignment, and I worked on it this summer at the Complex Networks Lab with Professor Tiana Milenkovic and PhD student Sean Gu. The first step of network alignment is typically graphlet counting, or describing the neighborhood of each node in terms of which subnetworks it belongs to. Here are all possible 2 to 4 node graphlets. Okay, straightforward enough. Advances in biological data collection technology have made things more interesting, though. Now we have an abundance of data in the form of heterogeneous networks that by definition include multiple types of nodes and edges. In response, the Complex Networks Lab has generalized traditional homogeneous network alignment into novel heterogeneous network alignment, which takes advantage of node or edge type information to generate a more accurate mapping. For my project, I modified the original heterogeneous network alignment method to include more information about the colors in nodes' neighborhoods. Here are some heterogeneous versions of one graphlet that are all counted separately in my exhaustive method. We're still in the process of testing, but it appears that exhaustive graphlet counting does not necessarily improve alignment accuracy. This means that we can continue to use the less computationally intensive, non-exhaustive approach without compromising accuracy. While developing exhaustive heterogeneous graphlet counting, I also developed a new homogeneous graphlet counting algorithm that can easily be extended to count heterogeneous graphlets, or even weighted graphlets. Perhaps you'll hear about those in a future student's video. Preliminary tests show that my algorithm, in yellow, is up to 10 times faster than its predecessor in green. In conclusion, I'm grateful to the Complex Networks Lab and the DISC REU program at Notre Dame for this opportunity to help network scientists take on some of the world's most important challenges, and thank you for watching.